pleasure for me to introduce our today's guests, Professor Błasiak and Dr. Aleksandra Trenk. So I would like to tell you a few words about them. Professor Błasiak is an accomplished neurobiologist, and her team work sheds light on how the brain processes novelty and responds to stress. What is worth mentioning is the fact that she leads a research group at the Jagiellonian University's Department of Neuropsychology and Chronobiology. And this team focuses on brainstem circuits, particularly the nucleus incertus, and their role in regulating many processes such as anxiety, memory, and reward-related behaviors. Professor Błasiak combines behavioral models with techniques, very popular and trendy, let's say, techniques nowadays, like optogenetics, chemogenetics, and ex vivo electrophysiology, to explore the nucleus incertus hippocampus axis and how it shapes contextual memory and emotional responses. She also received a lot of funding, including the National Science Center, and these projects are not only methodologically strong, but also highly relevant to understanding the mechanisms behind anxiety and PTSD. And beyond the research, which is a very high level, she serves as a director of the Institute of Zoology and Biomedical Research. And what is, I think, very important is that she supports young scientists, including through the student-led neuroscience club Neuronal. I'm sure that a lot of you heard about it. Today's seminar will be co-led by Dr. Aleksandra Trenk, a key collaborator, and they will highlight their joint interdisciplinary work and showcase how advanced tools help us dissect neuronal circuits and the action of neuroactive compounds. So without further ado, I'm really happy and delighted to have you with us today, and I would like to give the floor to you.
Thank you very much. Thank you for very, very nice introduction, Carolina, and thank you for letting us share our not only technique but also a little bit about our the topic we are trying to investigate. As you mentioned before, it will have a lot of information I will share with you. At the very beginning, we'll reflect the connection between the brainstem and the ventral hippocampus. So I will start with the introduction that will show you how we are trying to resolve the neuronal mechanism underlying very, very complicated brain functions. So let me share my screen with you. Okay, please let me know if you can see.
Yes, we can.
The first slide, perfect. Okay, thank you very much. So today, together with Ola, we will talk about the multi-electrode array recordings and how this technique is helping us to understand very, very complicated brain functions. We are especially interested in social interactions, and social interactions are very important for our human well-being, but not only human well-being, but also in all mammalian kingdom. Social interactions, the quality of these interactions is of highest importance. So what you can see here is with the rise in social adversity, also morbidity and mortality rises. And with the social advantage, the score is morbidity and mortality is very low. And very important element of social interaction control is oxytocin. Oxytocin is a neuropeptide synthesized in the brain of all mammalian species, and the major site of the synthesis is paraventricular nucleus in the hypothalamus. What you see here is a scheme of a rat brain sagittal scheme showing the localization of paraventricular nucleus. And here on the coronal slice, you can see that paraventricular nucleus, as it is named, lies at the border of the third ventricle. And because it acts in very different brain areas, it controls a lot of different functions, and different biological and physiological outputs of oxytocin action can be observed. And while we are talking about behavioral outputs, this is maternal behavior control, bonding formation, social interaction, very interesting for us, social memory, but also stress relief, trust, and sexual behavior. At the same time, we know that oxytocin controls milk ejection, pain relief, also stress alleviation mentioned before, as well as food intake and metabolism control, and such processes as bone mass regulation. So oxytocin is of highest importance in social contacts control and many other functions. But at the same time, social interactions are very, very sensitive to stress. And we know that stress influences social interaction on many different levels. And we know that with the increase in stress intensity, the negative social interaction score is observed. And with increase in stress sensitivity, we can observe increase in aggression, social avoidance, social fear, and so on. At the same time, high social competence that is accompanied by low stress level is combined with increase in the value of parental and pair bonding, mating, and affiliation, and as a consequence, positive social interactions. But how the stress information about stress gets to the oxytocin synthesizing neurons? There are different ways, but one of the very important one is through relaxin-free neuropeptide. Relaxin-free neuropeptide is synthesized by neurons in a rat and other mammalian species brain, and it is quite little known so far, but we are trying to give it more attention, and we know that within the brain there are at least 4 different areas that are the sources of relaxin-free. But the major one, when the majority of relaxin-free neurons are localized, is nucleus incertus in the brainstem. Relaxin-free and nucleus incertus neurons innervate, as in case of oxytocin neurons, a lot of different brain areas. And as you can see here, nucleus incertus fibers can be found in almost all possible areas of the brain. And that includes hypothalamus, when oxytocin neurons can be found. We know that nucleus incertus, relaxin, and relaxin receptors, RXFP3s, are responsible for control of stress response and social interactions. And these processes, control of these processes, is most important for us in our studies. We know that nucleus incertus innervates hypothalamus. And we also know that within paraventricular nucleus, quite a lot of relaxin-free receptors, RXFP3s, can be found. And what you can see here is a plate from Allen Brain Atlas showing expression of mRNA for RXFP3s within paraventricular nucleus. So in fact, I can say that paraventricular nucleus is one of the places in the brain with the highest expression of RXFP3. So it is very much sensitive to relaxin-free, and the synthesis of relaxin-free and its release is dependent on the stress level and perceived stress. Our studies showed that both oxytocin and arginine vasopressin neurons within paraventricular nucleus express relaxin-free receptors mRNA, and what you can see here as these black dots are mRNA, stained mRNA for relaxin-free receptors, RXFP3s on oxytocin and arginine vasopressin neurons within hypothalamus. At the same time, we know from electrophysiological studies, from patch clamp recordings, that these neurons, oxytocin neurons within paraventricular nucleus, are very, very sensitive to RXFP3 agonist. And what you can see here is a recording with application of A2, mentioned agonist, and outward current elicited by the presence of the drug within the recording chamber. And what is important is that vast majority in both females and males, vast majority of neurons in paraventricular nucleus is sensitive to this, to activation of this receptor. At the same time, we know that nucleus incertus itself, and what you can see here is a slide showing nucleus incertus in the brainstem, nucleus incertus neurons expressing oxytocin receptors. So these two structures are very much interconnected, and relaxin-free controls synthesis of oxytocin within paraventricular nucleus, but stays under control of this hormone. And what was very, very interesting for us was the fact that all relaxin-free neurons within nucleus incertus express oxytocin receptors. So this connection and this dependence is very, very tight. So we knew that these two areas of the brain, we know it, that PVN and nucleus incertus are connected and dependent from each other. So our guess is that paraventricular nucleus and neurons that synthesize oxytocin within this structure receive information about stress from nucleus incertus. At the same time, we ask the question, what might be the common structure that can integrate signal from oxytocin paraventricular nucleus, so signal that regulates and controls social interactions, and from nucleus incertus, so information that informs the brain, including PVN, oxytocin neurons, that informs the brain about the stress factors. And such a structure is ventral hippocampus, because we know that this area of the brain is involved in stress control, contextual fear formation, social interaction, and many other functions that still are under control of nucleus incertus and PVN at the same time. So we hypothesized that ventral hippocampus is a hub that combine this social salience information and stress information. And we had quite a lot of anatomical reasons to believe so, because both previously published data showed that within ventral hippocampus, you can see here ventral hippocampus of the rat in the rat brain, is very densely innervated by nucleus incertus fibers. And our recent data, when we injected AAV vectors driving expression of fluorescent YFP protein into nucleus incertus, and by doing so, we could map all the axonal endings of nucleus incertus neurons in the brain. These studies showed us that within ventral hippocampus, and what you can see here is a horizontal slide from ventral hippocampus, a very dense nucleus incertus fibers complex can be observed, and at the same time, these fibers were relaxin-free positive. And within ventral hippocampus, very specific area is innervated by nucleus incertus and relaxin free fibers. And this area is called dentate gyrus. And the dentate gyrus is kind of an entry gate to the whole hippocampal formation. So we knew that the fibers are there. We also knew from our multiplex in situ hybridization studies that within a rat hippocampus, especially within the dentate gyrus, there is quite a lot of neurons that express mRNA for RXFP3 receptors, so receptors for relaxin-free, and that these neurons were, in all cases, GABAergic ones. So they expressed VGAT1 mRNA. At the same time, we observed that in human brain, and this is the slice from human dentate gyrus, also RXFP3 receptors can be observed. And similar to rat brain, this expression was restricted to GABAergic neurons. This technique, this multiplex in situ hybridization, allowed us to also verify the type of GABAergic neurons within dentate gyrus, and we know that these are different kind of GABAergic neurons within dentate gyrus, including somatostatin and parvalbumin neurons. We also knew from our molecular studies that within the dentate gyrus, we can observe neurons, a population of neurons that at the same time express oxytocin receptor mRNA, shown here as blue dots, and relaxin-free mRNA, shown here as purple dots. So you can see here in the first and second row an example of neuron that express both type of receptors. But we also observed neurons that were positive only for one receptor, in this case only for oxytocin, and here for relaxin-free receptor mRNA. So we had like all the anatomical reasons to believe that indeed ventral hippocampus is a hub that integrates social salience information from paraventricular oxytocin neurons and stress information from nucleus incertus. But we had no functional data and no functional proof to say that the integration of these signals through oxytocin and relaxin-free indeed occurs within ventral hypothalamus. And to functionally verify the fact that this integration at the level of neuronal activity is present within ventral hippocampus, we engage multi-electrode array recordings to verify our hypothesis. And now I will give the floor to Ola, who will tell you how the technique is working.
Okay, so I will share my presentation with you. So I will introduce you to powerful methods we use in our research, but before diving into how multi-electrode arrays work, let's briefly review what electrophysiological recordings are. So the electrophysiological signal can be recorded intracellularly. The recording electrode is placed inside the neuron, and we can record changes in voltage, which gives us detailed information about its membrane potential. By using patch clamp techniques, which is a specialized intracellular method, we can use a glass pipette to form a tight seal with a cell membrane and record membrane potential or ion currents flowing through the membrane. And finally, we have extracellular recordings, and the electrode is outside the cell, and we can record electrical signals that neurons generate into the surrounding tissue. And today, we will focus on this type of recordings. By placing electrodes outside the cell, we can detect two complementary signals, action potentials and field potentials. Action potentials, or spikes, are high-frequency millisecond scale waveforms emitted when individual neurons fire. And field potentials are the combined electrical signals from many neurons that appear as slower voltage fluctuations in the extracellular space. Regarding action potentials, their shape will be different depending on the placement of the recording electrode. So when recorded intracellularly, the measured trace has its characteristic rapid depolarization and slower repolarization. Whereas when recorded extracellularly, the recorded waveform has smaller amplitude and reversed shaped. This is because we record the signal from different sides of the cell membrane. Okay, so now we can move to multi-electrode array recordings. Multi-electrode array, shortly MIA, is a flat chip with a well at its center. The well contains a regular grid of microelectrodes, and when performing an experiment, you place the tissue slice directly on top of the electrodes so that each electrode contacts the tissue. The well is filled with culture medium, or as in our case, with artificial cerebrospinal fluid, which is an ionic solution designed to mimic the brain's natural extracellular environment. The medium buffs the tissue and keeps it alive for many hours of recordings. The arrays of microscopic electrodes allowed us to measure the electrical activity of individual cells and neuronal networks. Many different matrices are available from which we can choose which will work best for our experiments. And the arrays can differ in the shape of the grid. So you can have square one, hexagonal, or even a custom layout. They can differ in the number of electrodes from a few dozen to several hundred. And they can differ in spacing between electrodes. The spacing will determine the spatial resolution and the area covered. Moreover, electrodes themselves can have different geometries. So flat electrodes are standard for general extracellular recordings, but for example, cone-shaped electrodes can improve contact with the tissue and recording quality. And for example, mushroom-shaped electrodes may enhance adhesion or be useful in specific 3D configurations. Additionally, you can use perforated MIAs, and these are the electrodes that we are using in our studies. This chip has micro-perforations in a regular grid around the electrodes, and these perforations allow the artificial cerebrospinal fluid to flow through the tissue, improving oxygenation and nutrient delivery. It's especially helpful for thick slices and for long recordings. And in addition, due to the perforation, we can have a better contact between the electrode and the tissue. Multi-electrode array systems are not limited to passive recordings. They can also be used for stimulations. And here the individual electrodes can be programmed to deliver controlled voltage or current pulses to the tissue. This led us to activate specific neurons or regions within the slice and study evoked responses across the network. MIAs can also be combined with optogenetic stimulations to target genetically defined cell populations, expressing light-sensitive proteins, for example, channel rhodopsin. This approach offers cell type specificity but there are some practical challenges such as precise light delivery or avoiding stimulation artifacts during recording. That is why this method is still being refined. Multi-electrode arrays are very comprehensive tools. They can be used to record the electrical activity of various types of cells, not only neurons, but also any other cell that exhibit electrophysiological properties, for example, cardiomyocytes. MEAs can be used in both in vitro and ex vivo preparations. In vitro studies, cell cultures or organoids are used. And here on the right side, you can see a human brain organoid. And we were able to record the activity of the cells. This model is especially valuable for applications such as disease modeling or drug screening. For ex vivo studies, brain slices are used, and here you can see a rat hippocampal slice placed directly on the electrodes of our multi-electrode array. This option allows us to record from neuronal circuits in well-preserved brain structures, and it's ideal for studying region-specific activity, network dynamics, or effects of pharmacological manipulations. For ex vivo experiments, we have to prepare proper slices. And in the first step, we have to collect the brain. So the brain is quickly removed from the skull and immediately immersed in ice-cold oxygenated artificial cerebrospinal fluid to minimize hypoxia and preserve tissue integrity. After that, the tissue is mounted on the vibroton stage and cut into 200 to 400 micrometer thick slices, also in ice-cold oxygenated artificial cerebrospinal fluid. And then slices are transferred to a chamber filled with warm artificial cerebrospinal fluid. And after at least 30 minutes of recovery, slices are ready for electrophysiological recordings. This slide shows a basic overview of the MEAS system setup. So firstly, we have the multi-electrode array where we put our tissue or cell culture and where the recording occur. MEA is connected to a head stage which amplifies the tiny extracellular signals. Then the signal goes to the interface board. It's further amplified, digitized, and transferred to the computer, where using proper software, we can record, visualize, and analyze the data in real time, as well as to program our stimulation protocols. Together, these components form the complete system for both recording and stimulating neuronal activity with high precision. On this scheme, you can see how our perforated MEAS system setup looks like. So you can see here all elements I have just described. So we have the head stage with dual recording capability. So in our case, our system allows us to record from two MEAs at the same time. Then we have the interface board, computer, and there is also a temperature controller to maintain proper conditions through the whole experiment. And there is also the whole tubing system to perfuse the tissue with artificial cerebrospinal fluid. For perforated multi-electrode arrays, we have two flows, the upper one and the lower one. And this allows the artificial cerebrospinal fluid to flow through the tissue. And additionally, the constant perfusion allows us to administer drugs here we have a system of special syringes where we can apply different substances. Okay, so now we can move to data acquisition. So here you can see an example of raw data being acquired from hippocampal slice. What you are looking at is a recording of extracellular signals from multiple electrodes simultaneously. In our case, it is 60 electrodes on one MIA, and we can record from 2 chips at the same time. Each box here represents a single electrode, allowing us to track the spatial and temporal patterns of activity. The fast, sharp changes represent action potentials, spikes, from nearby neurons, and one electrode can record the activity of a few neurons. This kind of data forms the basis for further analysis. And depending on whether we want to analyze spikes or slower activity like field potentials, we apply filters to distinguish these signals. So low-pass filter isolates the local field potential, which is a slower signal that reflects some synaptic activity and broader network dynamics. And high-pass filter extracts action potentials, which are the fast events generated by individual neurons. And this frequency-based separation allows us to analyze both single-cell activity and population-level processes from the same recording. Mostly, we are interested in action potentials, so we need to perform spike detection. On this slide, the traces are high-pass filtered, so action potentials stand out clearly. And the red line here indicates the detection threshold. Any crossing is counted as a spike, and those spikes are then binned in real time and plotted as histogram of spike counts. And this is providing an online view of firing rate dynamics. And in this example, you can see the response to oxytocin administration. So in this case, it's increase in firing rate. As I already mentioned, each electrode on TMEA often records action potentials from multiple nearby neurons, and this is what we call multi-unit activity. In real time, the histograms I just showed reflect the total activity of all cells detected at a given location. However, if we want to study the activity of individual neurons, we perform offline spike sorting by grouping waveforms, so we can isolate the activity of single unit from the mixed signal. This allows us to analyze the firing pattern of each neuron separately, revealing cell-specific responses that would be hidden in a broader multi-unit readout. So multi-unit activity gives us a quick overview of the local network activity, while single-unit activity shows us the dynamics of individual cells. There are several tools for spike sorting, and we are using KiloSort, an open source tool for recordings with large number of channels. And the pipeline involves 4 main stages. Firstly, we have pre-processing. The raw traces are filtered and corrected for slow drift in tissue. Then there is spike clustering, which is initial grouping of waveforms using amplitude-based model with automated splits and merges to refine clusters. After that, there is template matching. The spikes are separated by fitting them to evolving spike shape templates, ensuring accurate assignment. And finally, post-processing, it's final cluster refinement and unique unit classification to produce clean spike trains. After kilosort does its part, we have to refine the spike trains manually to ensure accuracy. And we inspect principal component analysis to confirm that each cluster is well separated. When clusters overlap, we may split or reassign spikes. By examining auto and cross colorograms, we verify that each unit's firing pattern is physiologically possible. And with the review of average spike waveforms across channels, we ensure consistency in shape and amplitude. Any outliers are removed or reclustered. And through this manual selection, we refine the automated output into a trusted set of data for further analysis. And the final step is data visualization. We can prepare spatial heat maps encoding the multi-unit activity for the whole recorded area. So the gray dots here represent spots of multi-electrode array, and the change in activity after administration of the substance is color-coded. So the yellow and red colors here indicates excitation. We can also visualize single unit activity by preparing a temporal heat map. And each line here represents single unit. The activity was normalized and expressed as a value from 0 to 1. The red color indicates higher activity and the blue decrease in firing. And we can see responses to drug application. And in this example, you can see a nice inhibition after application of our agonist. We can also have a look at each neuron individually. And here you can see an exemplary neuron recorded during the drug administration. On top, you can see separated spikes of a single neuron, and on the bottom, the corresponding frequency histogram. The shaded rectangles indicate the duration of the action of the drug. And finally, we can reconstruct the location of the recorded neurons. As in this example, you can see where were located the cells that were inhibited, shown in blue, and where were located cells that were excited by our substance. Recordings using MEAs are powerful techniques, as I showed. But as with any method, they come with both advantages and limitations. And the pros include the fact that we can record the activity of tens to hundreds of neurons simultaneously. This helps us reduce the number of animals used. We can analyze the local neuronal networks. So MEA reveal not just single cell spikes, but also how groups of neurons interact over space and time. And we can exactly see when and where the activity changes. We can map the effect of the drug that we test. We can also record and stimulate the tissue using the same device. And moreover, with proper perfusion, we can record from the same slice or cell culture for hours or even days. And finally, conditions like temperature, perfusion, stimulation protocols, etc., all are precisely controlled. As for the cons, by using slices, we cause mechanical damage to the tissue. In acute slices, long-distance connections are disrupted, so we miss the whole brain dynamics. And also, the standard planar MEAs record predominantly from the tissue's topmost layer. And even under optimal conditions, slices remain alive for a limited period. And what is worth noting is that large recordings with many channels generate huge data sets that require significant storage capacity. And finally, high-density arrays and amplifiers are very expensive, and the data analysis demands expertise. Still, multi-electrode arrays have many applications. They can be used for drug testing. They enable efficient pharmacological research by monitoring how different drugs modulate firing rates, activity patterns, or network synchrony. In cell cultures and organoids, MEAs track spontaneous activity, emergence, and maturation, revealing how connectivity evolved during development. Furthermore, by combining electrical stimulation with recording, MEAs can induce and measure plasticity processes like long-term potentiation at multiple locations. Also, patient-derived neurons or disease-specific organoids and their recordings using multi-electrode arrays help us to study abnormal network dynamics in different neurological conditions, for example, in epilepsy, Alzheimer's, or Parkinson's disease. And with the long-term stability, multi-electrode arrays can record daily oscillations in neuronal activity, offering insights into how the intrinsic clocks regulate the network behavior over the 24-hour cycles. And that's it from my side. Now I'm giving the floor back to Ania.
Thank you very much, Ola. Now, I hope you will understand the results I would like to present you and how we answer the questions if indeed ventral hippocampus can be considered a hub combining, integrating social salience and oxytocin-driven controlled signal from paraventricular nucleus and stress-related and relaxin-free controlled information about stress. So as you can expect, we apply the techniques to record activity of ventral hippocampus from rat brain slices. And our recordings showed very nicely that application of oxytocin to ventral hippocampal slices is causing an increase in neuronal activity of ventral hippocampus. But what was very important for us was the observation that in the presence of relaxin-free, the response to oxytocin is diminished. So we first verified if the observed diminished response was not the effect just of the second application of the drug. In first experiments, we applied the drug twice, and we did not observe significant decrease. We did not observe significant decrease in the responses, but in the presence of relaxin-free, oxytocin was less powerful. And what you can see here is multi-unit activity recorded from our brain slices, and these are responses to 2 consecutive applications of oxytocin. And here, during the next recording, we applied oxytocin in standard ACSF and in the presence of relaxin-free. So the purple bar represents the presence of the drug relaxin-free in the recording chamber. And we observed that the response to oxytocin in the presence of relaxin-free is diminished. And we could observe it. Let me show you the pointer. We could observe it both at the multi-unit activity level and at the level of single neurons. Here, what you can see at the bottom traces, we can follow the activity of single neuron in response to oxytocin, and then in response to oxytocin in the presence of relaxin-3. And here is just another example showing very much the same. So, as I've mentioned before, from our molecular studies from in situ hybridization, we knew that within the hippocampus, within the dentate gyrus, there are neurons that co-express oxytocin and relaxin-3 receptors, but also there are neurons that express only one type of receptor, either only oxytocin receptor or relaxin-3 receptors. So to answer the question, if we are dealing in our electrophysiological recordings with direct response of specific cells to applied drugs, or is it indirect one? We had to prepare a mix of different drugs to answer this question. Why is it important? Because within the neuronal network that is preserved to some extent, as Ola mentioned, within brain slices, we can deal with situation when our applied drug directly activates postsynaptic receptors. And this activation may depend on presynaptic activity. But we also can deal with the situation when our drug can act on presynaptic receptors. So in this case, we are describe the activity we record or change in activity as indirect effect. So we just wanted to verify if the neurons we recorded from were directly, postsynaptically, or presynaptically activated by our drugs, by oxytocin and relaxin-3 respectively. To do so, we had to enrich ACSF with so-called synaptic blockers. So with addition of CNQX and DLAP5, which are antagonists of glutaminergic ionotropic receptors, and gabazine, which is an antagonist of GABA-A receptors, we could exclude the possibility that indirect response is important in our recordings. So what we did, we, as I mentioned, enriched the extracellular fluid ACSF with these synaptic blockers, and again applied oxytocin and relaxin-3. And similar to previous recordings, we observed that the same neuron in the presence of these blockers were able to respond to both oxytocin, shown here as a blue bar, and relaxin-3 shown as a purple one. So these drugs were still acting in the presence of synaptic blockers, but we could very clearly observe that these responses were much smaller than responses in control ACSF. So this data allowed us to conclude that we are dealing with both direct and indirect action of used drugs. So when it came to interaction between relaxin-3 and oxytocin, and defining the level of this interaction at the single cell level, or maybe more accurately at a molecular level, what we knew is that RxFP3 activation may lead to inhibition of neuronal activity by inducing an outward potassium current through so-called M-type potassium channels, KC and Q2. At the same time, other studies showed that very similar situation can be observed in case of oxytocin receptor activation, also in the hippocampus, but in other hippocampal area, CA2 field. So what was observed was the fact that oxytocin receptor activation also involved, the effect of this activation also involves KCNQ channels, but in this case, unlike in the case of RxFP3, we are dealing with reduction of potassium current flowing through these channels. So we figured out that what we can deal with is the situation that oxytocin, through closing KCNQ channels, can depolarize neurons and activate them within hippocampus, and at the same time, relaxin-free activation of RxFP3 receptors can act to activate these channels and hyperpolarizing, inhibiting neurons within hippocampus. And this could be potentially the neuronal and molecular mechanism of observed interaction between oxytocin and relaxin-free within hippocampal formation. So what we also knew from our molecular studies is that within hippocampus, there is expression of KCNQ2 mRNA, and this expression is colocalized with oxytocin receptor mRNA expression and relaxin-free mRNA expression. So again, we had all the anatomical reasons to believe that it might be the proposed mechanism, molecular mechanism, might be responsible for previously showed interaction between oxytocin and relaxin-free action within hippocampus. So using MIA recording, we showed that activation of oxytocin receptors with specific agonist of these receptors, drug called TGOT, induces very nice excitatory responses. And these are just 2 consecutive responses in the standard ACSF. At the same time, when we applied to the recording chamber specific antagonist of KCNQ channels, we observed that this response, this excitatory response, was abolished. At the same time, when we applied to a different slice, but also while recording hippocampal dentate gyrus activity, when we applied relaxin-free receptor agonist A2, we observed very nice inhibitory responses, but these responses were blocked by blocking the KCNQ potassium channel. So using MIA, we could conclude that RxFP3s and oxytocin receptors are expressed in a subpopulation of GABAergic cells in rat and human DG. Sorry, this conclusion was not drawn from electrophysiological recordings. We knew it from our molecular studies, but we knew using MIA that activation of these receptors exert opposite effects in a specific neuronal population within hippocampal formation, within dentate gyrus. And we knew that opposing interplay of these receptors may involve the same intracellular pathways. So we had not only molecular and anatomical data, but also functional data based on our electrophysiological recordings that support our hypothesis that ventral hippocampus is indeed a hub integrating social salience and stress-related information that is conveyed through paraventricular nucleus and oxytocin and nucleus incertus and relaxin-free to this area of the brain. So I hope we did convince you that multi-electrode array recordings is a powerful tool for exploring neuronal activity and the action of different neuroactive substances within brain slices, and also is a very, very powerful tool for exploring also intracellular mechanisms underlying different drugs action. And I would like to finish with that, and I'm happy to answer any of your questions.